Sorry, quite a wordy title. First of all, just introduction. My name's Ed Jarman. For anyone who doesn't know me, I'm a postdoc in Luke's lab up in Edinburgh at the Institute of Genetics and Cancer. What I really want to talk about today is some of our ongoing research looking at this interface between liver damage and inflammation and tumour initiation. And so this comes down to this concept that has been spoken about already today and yesterday, which is that risk factors for cholangiocarcinoma are often or always inflammatory or liver-damaging agents or conditions such as primary sclerosing cholangitis, hepatitis, liver fluke infection, etc. What's not really understood very well is how these regenerative and inflammatory cues coincide with genetic perturbations when tumours are initiated, because not all of these inflammatory cells become, not all of these biliary cells in the inflammatory environment become tumours. So something else has to be going on. The way we're doing this is by trying to model in genetically engineered mouse models this co-requirement for liver damage or inflammation and genetic perturbation. So because it's quite important for some of the slides that I'm going to present in this talk, I'm going to spend a little bit of time just explaining the mouse models we're using in detail, so I hope it's accessible and makes sense. First of all, we can lineage trace biliary epithelial cells using this keratin 19 CRE. So we have mice where we can treat them with tamoxifen, and this induces recombination of this lock-stop-LOX cassette, leading to a D-tomato, which is a bright RFP. Basically what this means is that when we treat these animals with tamoxifen, we get RFP expression specifically in cholangiocytes in these cells. With this otherwise wild-type animal, we can give them thioacetamide, which is a liver-damaging agent that Luke mentioned previously, and watch over a number of weeks. This is a 3-week time point where we see the initial signs of inflammation and liver damage and ductal regeneration. But in this model, we don't get any cholangiocarcinoma, we don't get tumours forming. So this is just a model for liver damage. Adjacent to this, we have this slightly more complex genetic mouse model where we include the deletion of P53 and P10. So here, when you treat with tamoxifen, you get this lineage tracing where you get RFP expression in cholangiocytes, and you also get the cholangiocyte-specific loss of these two potent tumour suppressor genes. So the reason we like these models and the reason we're interested is because you don't get tumours when you treat with damage alone. You don't get tumours when you treat these animals with tamoxifen and let them go about their business. But when you treat these animals with tamoxifen to cause tumour suppressor loss and then induce damage, over the course of 3 weeks, you get the formation of these early, very well differentiated cholangiocarcinomas. So just to explain what I'm showing here, these are just H&E sections at the 3-week time point for these two animals after liver damage. RFP expression shown in red, showing the ductal morphology of these tumours. And this is just a 3D tissue clearing example to show how these structures look in 3D. So the important concept here is we have a mouse model where we can separate out the role of damage and tumour suppressor loss and look at how these two things combine at the very early point of tumour initiation. So this is all in a 3-week time point where we first see these tumours emerge. So what we've run is a large-scale single-cell sequencing experiment to try and pick apart what the transcriptional events are in these contexts and define what role damage versus tumour suppressor loss is playing. So in this experiment, we've got 3 treatment groups. These are damage alone, which is our control animal with thioacetamide, P10 and P53 loss alone, which is our tumour suppressor loss animal after induction without liver damage. And we have what I'm going to refer to in this talk as the tumour permissive state, which is when we have these animals and we've both knocked out the tumour suppressor genes and also induced liver damage with thioacetamide. So when I say tumour permissive state, this is what I mean, this co-occurrence of damage and tumour suppressor loss. And what we've done is we've treated these animals for 3 weeks with the thioacetamide. We've FACS sorted out the RFP positive cholangiocytes. So an important part of this experiment is it's specifically on the biliary epithelial cells in these tumours, or the keratin 19 expressing cells in these tumours. We've run this through a standard 10x genomics single-cell sequencing protocol. The details of that aren't too important. Suffice to say that we have 10 different mice from these groups with over 50,000 cells sequenced at around 50,000 reads per cell. So we've done relatively deep sequencing in a large number of cells. And after data processing and quality control, we end up with a data set where we map the transcriptional landscape of over 20,000 cells from 10 mice in these 3 groups. So we can make a good comparison of what's going on between these 3 situations. This is a UMAP representation of all this data combined, just coloured by clustering, so a broad transcriptional identity. For anyone who's not familiar with this type of plot, each one of these points represents an individual cell, and where it sits on this X and Y axis is representative of its overarching transcriptional identity. It's probably the simplest way of putting it. We see it separates into 8 clusters. We can kind of, based on the genes expressed, we can identify what these are. The broad brush of this isn't too important, so the specifics of this aren't too important. There's a lot of kind of peripheral smaller clusters which we've ruled out as not hugely interesting. What we're going to look at today is these main clusters in the center here, including this tumor-specific cluster. And just to kind of orientate everyone with what we think these cells are, we have small duct cholangiocytes make up the mass of this data set in this 0. Cluster 5 is a proliferative cluster. I'm going to talk about these the most today. 1 and 2 are these large duct cholangiocytes, have all markers for larger ducts. And we have this tumor-specific group at the top right. So importantly, what matters is how this data looks when you look at the individual groups. So the damage-alone mice, the tumor suppressor loss, and the tumor permissive group, which is once again damage and tumor suppressor loss. You see that they don't perfectly overlap. They map differently to different clusters within this UMAP. So this suggests that there are interesting transcriptional differences between these groups that will allow us to unpick exactly what's going on. So if you overlay these like this, with green being the tumor permissive state, same as up here, you can see, once again, this kind of lack of overlap, which indicates what are the interesting processes that we can dissect out and look at to understand what's going on. I like to break this down into 3 processes that are interesting to look at. One is this transcriptional response to damage, where you see kind of the top of the UMAP is lined in this tumor suppressor loss alone, so without damage, but in the 2 damage groups, you see a shift downwards. And the important thing here for this talk is that this includes the emergence of this proliferative cluster. So this is a cluster that occurs in response to damage, which isn't too surprising. Another factor of this data is this shift at B, which is a P10 and P53 specific shift that seems only to be in the large duct cholangiocytes. This isn't tumor specific. We see it with and without damage, so we even see it in mice where we don't have tumors. So we're not entirely sure what's going on here, but we think this is quite interesting. I'm not going to focus on this today because we don't have all the answers, but it's an interesting piece of data that we can dissect out in the future. And most importantly, what we see from this is the emergence of cell clusters specific to the tumor permissive environment. So that's these green areas here. And these represent what we're looking at, the novel populations that only occur when tumors are allowed to form at this very, very early stage of tumor initiation. We can dissect out transcriptionally how these cells identify. So as I've said, today I'm going to focus on this proliferative cluster 5. These are just expression representations of expression for 2 very common proliferative genes, Ki67 and TOP2A. Other genes could have been shown, but this just demonstrates that this cluster is very specifically proliferative. So if we zoom into this and we look at it between our treatment groups, our damage alone, our tumor permissive group, and our tumor suppressor loss, as I pertain to, this is a damage-specific cluster. We only see this in these 2 groups here. It's very poorly represented in the tumor suppressor loss alone group. And if we overlay, excuse me, if we overlay transcriptional profiles for these, for this cluster, we see that actually there's a shift in transcriptional space, so where these cells sit when you look at the tumor permissive group. So what this means is that cells that are proliferating in response to damage in the environment of P10 and P53 loss are transcriptionally different to cells in damage alone, so the cholangiocytes when only damage is induced. So obviously we want to pick out what the differences are in the transcriptional profiles between these 2 genes to understand why the tumor permissive proliferative cells might be leading to tumor initiation. And if we look at, in a broad brush approach, KEGG pathways or gene ontology, actually what we see is a lot of immune signaling changes, which is quite interesting. And one of the ones I'm most interested in, which has been teed up perfectly today, is interleukin 17. We see an interleukin 17 signature specifically in proliferative cells in the tumor permissive state that we don't see when the liver is damaged on its own. In terms of gene ontology, we also see a positive regulation of wound healing, upregulated, once again, upregulated more than just the damage alone group. So this is a tumor permissive upregulation of regenerative and wound healing pathways. If we look at, so these are a number of genes that are downstream transcriptional targets of interleukin-17 signaling that, as I said, I'm particularly interested in. And we see that these are not only expressed in this right-hand side of our proliferative cluster, but we also see them in the other tumor-permissive populations that occur if we go back to this overlay here. So we've got these three tumor-permissive populations, and they seem to share a transcriptional identity in terms of upregulation of immune signaling, including interleukin-17. And what we believe is happening is that these tumor-specific groups are a result of this aberrant proliferative population here. So we've got a tumor seeding group here at the very early stages of tumor initiation that occur as a response to damage, because this proliferative group is only induced when the liver is damaged. If we look at this in a bit more detail and look at individual genes that might be interesting, we've actually noticed that, once again, in this tumor-permissive group in the proliferative cluster, we see a specific upregulation of the genes Wnt and Wnt7b. So Wnt is a promoter of Wnt ligand secretion. So these two together suggest quite strongly that there is a change in Wnt ligand secretion, specifically in these proliferative cells when P10 and P53 are lost. And this is just the quantification of gene expression values between these two groups for these two genes. So we can go back into the tissue. This is the joy of working with mouse models, is that we have a lot of access to as much tissue as we need, and we can look at things in situ. So if we look in our tumor-permissive group, this is co-immunofluorescent staining for the RFP, which is the lineage tracing of cholangiocytes, and Wnt7b, which I've shown is transcriptionally upregulated. And we see small specific populations within the tumor mass that express high levels of Wnt7b. Most importantly, we don't see these in damage alone. We don't see any evidence of cholangiocytes expressing Wnt7b, nor in the tumor suppressor loss alone group. So we have a novel population of Wnt-secreting cells at the very early stages of tumor initiation. So what do we think is happening? I've taken some cues from other systems, and other publications. One thing we've seen in the literature is that P53 has been shown to be sufficient to drive Wnt ligand secretion in this example, breast cancer cell lines. So actually, it might be a direct result of P53 loss that these early proliferative cells are expressing Wnt ligands. So what we suspect is that P10 and P53 mutations in these cells are promoting a Wnt secretory phenotype specific to this damage-induced proliferative population. So this can be a potentially first point at which this intersection of damage and tumor suppressor loss leads into a novel tumor-promoting phenotype. We need both the damage to drive proliferation and the P10 and P53 loss to cause this aberrant proliferative phenotype with Wnt secretion as a feature. What we suspect, based on the idea that we see interleukin-17 signatures and other immune regulatory signatures throughout our tumor-permissive group in different cell clusters, is that actually this Wnt secretory phenotype might be promoting an interleukin-17 high inflammatory signature. I'm not an immunologist, but what I understand from the literature is that this has been shown in this breast cancer paper, actually, that Wnt secretion is sufficient to drive neutrophilic inflammation through interleukin-17b. We currently don't know what the signal receiving cells are for this Wnt signal, but one suggestion is that they might be macrophages, which have been shown to respond to Wnt ligands and promote the secretion of interleukin-1b, which can be an initial part of this cascade leading to this interleukin-17 high tumor-initiating environment. And so what we suspect is that this high interleukin-17 signaling might be acting directly on these mutant epithelial cells, because we're seeing transcriptional signatures pertaining to interleukin-17 within these tumor cells to establish an unbridled wound healing response, because interleukin-17 signaling is extremely important in wound healing and regeneration at mucosal barriers. So we think these might be the first pathways that these cells are perturbing in response to tumor suppressor loss. So I think in summary, we've created this large single-cell transcriptional data set, which is really allowing us to unpick the importance of damage versus tumor suppressor loss. It's early days, we've still got a lot of analysis to do, and we still need to go back into the tissue to fully understand exactly what's going on for a lot of these questions, but we now have this really, really valuable resource in this large-scale data set. We think that tumor suppressor loss is inducing alterations in transcriptional profiles, specifically in these proliferative populations, as I've said, and this could represent an initial action of these cells that coincides at the interface of damage and tumor suppressor loss. And we see this interleukin 17 signature at the very early stages of tumor initiation. So we think this might be important in allowing these, I think one thing that's important to stress actually here is that at this stage of tumor initiation, these are well-differentiated cells. They're not hugely aberrant late-stage cancers. So we're seeing changes in these immune signaling pathways at a very early stage, and this might be an aberrant wound healing response that leads into cancer in the future. So what we're hoping is that by characterizing and using these models, we can understand what some of these very first events are in tumor initiation, and this might be really important for biomarker for progression into cancer in patients with things like PSC, or also potentially the development of new therapeutics for really early intervention. Cool. And with that, I'll just say a quick thank you to everyone in the lab and everyone who's helped with this work. It's a big team. And thank you to the AMF for the opportunity to speak about my work, and to you guys for listening. Thank you.